There is a silent pandemic unfolding across the globe. It doesn't grab daily headlines like a viral outbreak, but its toll is staggering. In 2019 alone, it claimed an estimated 1.27 million lives, more than HIV, AIDS, and malaria combined. This is the crisis of antimicrobial resistance, or AMR. It's a future where a simple cut could become deadly, and routine procedures like surgery or chemotherapy could become too risky to perform. For decades, our defenses have been crumbling as bacteria evolve faster than we can create new drugs. The pipeline of new antibiotics has been running dry. But now, from the front lines of scientific innovation, a new hope is emerging, a class of powerful new drugs dubbed super-antibiotics, designed to outsmart the most dangerous superbugs on the planet. The human cost of AMR is not an abstract statistic. It's the story of people like Vanessa Carter, who survived a horrific car accident only to battle a relentless MRSA infection that no conventional antibiotic could touch. It's the story of Mallory Smith, born with cystic fibrosis, who ultimately lost her life to a lung infection that had developed resistance to every single available medication. These stories are a terrifying glimpse into a potential post-antibiotic world. So why did we get here? For years, the scientific and financial challenges of discovering new antibiotics led most major pharmaceutical companies to abandon the field. It's expensive, it takes over a decade, and the return on investment is low because new antibiotics are rightly used sparingly to preserve them. This created a dangerous void in our medical arsenal. But a recent surge of breakthroughs, powered by new technology, is finally beginning to turn the tide. Let's meet the new arsenal. First up is Zosurabalpin. This is a huge deal because it targets one of the most feared pathogens, carbapenem-resistant Acinetobacter baumannii, or CRAB. The World Health Organization lists CRAB as a priority 1 critical threat. It's a major cause of hospital-acquired infections, especially in ICUs, leading to deadly pneumonia and sepsis. Gram-negative bacteria like CRAB are notoriously hard to kill because they have a protective outer membrane, like a fortress wall. Zosurabalpin has found a new way to breach that wall. It works by blocking a transport system that moves a key building block, called lipopolysaccharide or LPS, to that outer membrane. Without this supply chain, LPS builds up inside the bacterium, causing the cell to burst. It's an entirely new mechanism of attack, meaning existing resistance tricks don't work against it. Zosurabalpin has already succeeded in mouse models and is now in human clinical trials. Next we have Clovibactin and Teichobactin. These were discovered in a place scientists had long overlooked: soil bacteria that were previously considered unculturable. These antibiotics are masters of attacking gram-positive bacteria, including notorious superbugs like MRSA. Their strategy is brilliant. They don't just target one thing. They bind to multiple essential precursor molecules that bacteria need to build their cell walls. Clovibactin goes a step further. It actually self-assembles into large fibrils that wrap around these molecules, trapping them like a net. Imagine trying to build a brick wall, but someone keeps stealing all your different types of bricks and mortar. Because they target multiple highly conserved parts of the bacterial machinery, it's incredibly difficult for bacteria to evolve resistance. In fact, in laboratory studies, scientists have so far failed to generate any resistance to Clovibactin. That's almost unheard of. But what about the side effects of antibiotics? Broad-spectrum drugs often act like a bomb in our gut, wiping out the good bacteria along with the bad. This can lead to nasty secondary infections. Enter Lolamicin, a potential smart bomb. Lolamicin is designed to be highly selective. It specifically targets a transport system found only in gram-negative bacteria, the very group that includes pathogens like E. coli and Klebsiella pneumoniae. It shuts down their ability to maintain their outer membrane. But here's the clever part. It was designed to spare the beneficial bacteria in our gut. In mouse studies, Lolamicin successfully treated life-threatening pneumonia and sepsis without disrupting the gut microbiome. This prevented the secondary infections that often follow a course of powerful antibiotics. It's a new blueprint for developing smarter, more targeted drugs. And the good news keeps coming. Gepotidacin is a first-in-class antibiotic that was recently approved for treating uncomplicated urinary tract infections, or UTIs. This is the first new oral antibiotic for this incredibly common ailment in nearly 30 years. It works by inhibiting two enzymes essential for bacterial DNA replication. Other antibiotics target these enzymes, but gepotidacin binds to them in a completely new way, allowing it to work even against strains that are resistant to older drugs. It's a desperately needed new option for doctors on the front lines. So where are all these new ideas coming from? A huge part of the answer is artificial intelligence. Searching for new antibiotics used to be like looking for a needle in a cosmic haystack. Scientists would have to manually screen thousands of compounds. Now AI can do it in a fraction of the time. Researchers can train machine learning models to recognize the chemical structures of molecules that might have antibacterial properties. They can then unleash this AI on vast digital libraries containing millions or even billions of compounds. This is how a new candidate called Zosurabalpin was discovered. An AI model was specifically trained to find compounds that could kill Acinetobacter baumannii. It sifted through thousands of molecules and pinpointed Zosurabalpin as a potent killer. Lab tests confirmed it. AI is not only speeding up discovery, it's helping us find fundamentally new types of weapons we never would have found otherwise. But finding new drugs is only half the battle. We have to remember that bacteria are masters of evolution. They have an incredible arsenal of defense mechanisms. Some produce enzymes that literally chew up antibiotics. Others can change the shape of the antibiotic's target so the drug can no longer bind to it. Gram-negative bacteria can shut the door by closing the pores in their outer membrane, and many bacteria have developed efflux pumps, tiny molecular pumps that actively spit the antibiotic out of the cell before it can do any harm. This is why multi-drug resistance is so common. So even with these new super-antibiotics, if we continue to misuse them, resistance is inevitable. The race is never truly over. This brings us to the economic problem. Why did the antibiotic pipeline dry up in the first place? It's what experts call a broken market. It can cost over $1 billion to develop a new antibiotic, but the financial return is minimal. Because new drugs are used sparingly to preserve their effectiveness, sales are low. It's a terrible business model. To fix this, new ideas are being put into action. The first are push incentives. These are grants and public-private partnerships, like an organization called CarbX, that help reduce the cost and risk of the initial research and development. They push innovation by funding the early uncertain stages. The second, and perhaps more revolutionary, are pull incentives. These aim to create a viable market at the end of the process. The most talked about idea is the, quote, subscription model, sometimes called the Netflix model for antibiotics. Instead of paying per dose, governments or healthcare systems would pay a fixed annual fee for access to a new antibiotic, regardless of how much is used. This decouples a company's profit from sales volume. It guarantees a return on investment, making development worthwhile, while also encouraging stewardship, since there's no financial pressure to sell as much as possible. Finally, we can't talk about fighting AMR without talking about diagnostics. Developing new drugs is crucial, but preserving the ones we have is just as important. Rapid diagnostic tests are a game changer. First, they can quickly tell a doctor if an infection is bacterial or viral, preventing the useless and harmful prescription of antibiotics for a cold or the flu. Second, they can identify the specific pathogen causing the infection. This allows for targeted therapy with a narrow-spectrum antibiotic instead of a broad-spectrum one that damages our microbiome. And third, advanced tests can determine an infection susceptibility profile. Essentially, which antibiotics will work and which won't. Getting this information in hours instead of days means patients get the right drug from day one, improving outcomes and reducing the chance for resistance to emerge during treatment. Integrating smart diagnostics with smart prescribing is essential to safeguarding our new super-antibiotics for the future. The emergence of Zosurabalpin, Clovibactin, Lolamicin, and other super-antibiotics represents a powerful ray of hope. These innovations, driven by new science and AI, have the potential to save millions of lives and protect the very foundations of modern medicine. But they are not a silver bullet. The specter of resistance is always there. To secure a future where our medicines continue to work, we need a global, multi-pronged attack. We need to embrace sustainable financing models to keep the R&D pipeline full. We must invest in and deploy rapid diagnostics to ensure the right drug is used every single time. And we all have a role to play in stewardship, from doctors to patients, using these precious resources wisely. The war on superbugs is a marathon, not a sprint. These new drugs have given us a critical advantage, but our collective responsibility will determine whether we win. The future of medicine depends on it.